Welcome, students, to Chapter 4, which is a tour of the cell. So we're going to talk about the smallest unit of life, the cell. But first, a little history. For kind of most of recorded human history, we were pretty unaware of the microscopic world. Our naked eye does not process things that are microscopic, super tiny, and so it wasn't until we really developed microscopes in the mid-1600s-ish, and then successively better and better ones, that we really got to kind of expand our view of the world and see all these tiny microscopic organisms that live in pond water, for instance, or even just the inside of our own bodies. It started with Robert Hooke, mid kind of 17th century there, and he was using some pretty basic microscopes with just a little bit of magnification, not really a whole lot of sophistication to it, but he looked at the bark from an oak tree, and it looked to him like the bark was composed of a bunch of tiny little rooms, and the word for that in Latin happens to be cellulae, and so that's the term that we now use to refer to the kind of microscopic parts of our tissues, cell. A little bit later in the 1600s, Van Leeuwenhoek looked at blood and sperm. I think he was the first to actually look at and see human blood. He also looked at pond water and kind of all the amazing kind of protists and other kind of little microscopic organisms that exist in the water. He was using slightly more sophisticated microscopes than Hooke, and they kind of just built back and forth on each other and worked with kind of to expand and to look at this amazing kind of just microscopic world that heretofore we did not know about. And that's what really this chapter is going to focus on is this. The cell. So we'll talk a little bit about kind of what cells look like, how they function, so on and so forth. So this is kind of how this chapter is set up. So we're going to start with a little bit of an introduction to the cell, kind of compare cell types. Then we'll talk about the nucleus and ribosomes, kind of genetic control of the cell. Then we'll talk about how the cell makes stuff using the endomembrane system, and then how cells get energy, and the two organelles that help us do that. And then we will end with the cytoskeleton and some cell surfaces and how they might be a little different from each other. So this lecture here will actually be the first in a series of lectures about the kind of different parts of the chapter. So we're going to start with a little bit of an introduction. So in the lab, we use light microscopes. And if we want something a little more detailed, then we need electron microscopes, either a scanning electron microscope or a transmission electron microscope. So there's really kind of two concepts when it comes to microscopy: magnification and resolution. Magnification has to do with the size of the object. So we're like zooming in, right? Like you're doing a little, when you put your fingers on the screen and you pinch it together and you push them apart, right, and you zoom in, that's magnification. And then resolution has to do with how clear the image is, right? And you really need both of them for microscopy to work, right? If you've ever looked at sort of a low res image and you zoom in really far, it's all really pixelated, right? It's not very clear. So we need both high resolution and high magnification to kind of go hand in hand with each other. So the three images you see here, the one on the left is with a light microscope. So we've zoomed in just a little bit, 230 times bigger than its normal size. The middle image is with a scanning electron microscope, so slightly bigger magnification. It has been colorized using some computer algorithms. And then lastly, there's a transmission electron microscope, and if you look at the magnification on that sucker, we're talking about 9,000 times the size of the object. And so essentially what we're doing is we're zooming further and further into these different objects. The electron microscopes have much higher magnification and much higher resolution, and so if you really want to see sort of inside a cell, and see some of the kind of machinery of the cell outside of the nucleus, then you really need a more high-res, high magnification microscope. But the light microscopes that we use in lab are great for displaying cells, both kind of living and preserved. We can see the shape of them, we can see their nuclei, how they interact with each other. So the light microscopes are still incredibly useful and powerful tools to study biology. So when it comes to studying the cell, we always really start with something called cell theory. And this is an idea that was developed kind of mid-1800s. We'd had, about a century or so, 2 centuries or so of improving microscopes and kind of sussing out scientific methods. And it's this idea that essentially the cell is the smallest unit of life, right? So all of those things that we talked about in the very first chapter, metabolism, responsiveness, digestion, growth, development, reproduction, all of those kind of characteristics of life are really actually carried out by the cell. The cell is the smallest thing that can carry out those kinds of processes. So smallest unit of life is the cell. That means everything that is living is composed of cells. Some organisms are single-celled, so unicellular, just one tiny cell. Others, like us, are multicellular, meaning they are composed of many cells. Now, cells do not arise spontaneously. They do come from other cells. And even in our evolutionary history, the more complex cells that are of which we are composed developed from simpler cells. So let me ask you a question. Let's say we had 3 different things that we want to view using microscopes. The shape of a human white blood cell, the fine details on the surface texture of a piece of human hair, and then what about the detailed structures of an organelle inside of our liver cell? Which microscope do you think we would need, or which kind of microscope do you think we would need for each of those? Give you a minute to think about it. So if you said that we could look at the human white blood cell with a light microscope, you would be correct. So you'll see our kind of unaided eye, our naked eye. Of course, we can see height for humans. Everything from about 10 meters to, about 100 microns. We really have no problem kind of processing. From about 1 millimeter to about not quite 100 nanometers. We can use these light microscopes, but if we really want to see smaller things like the organelles inside a cell, or even the proteins inside a cell, then we are going to need one of these electron microscopes. A lot of bacteria are visible using light microscopes. In fact, most of them are visible using microscopes, but the very smallest bacteria or viruses, like if we want to see the spike protein on the SARS-CoV-2 virus, we would need an electron microscope in order to do that. We certainly can't do it with a naked eye, and even the light microscopes are definitely not going to give us enough magnification or enough resolution to see those smaller pieces of a cell. So since cells are microscopic, they have this very large surface to volume ratio. And eventually, I don't know if you guys ever watched a building implode. I love watching buildings implode. There's something amazing about kind of watching it collapse in on itself. Essentially, that's what would have happened to kind of the ancient cells in our evolutionary history, because they kept getting bigger without having, and they kept getting bigger and bigger and bigger and bigger, but still a really tiny surface area. Right, so if you look at the single kind of cube here, it's got a volume of 27 units cubed and a surface area of 54 units squared, right? So little tiny, relatively small surface area compared to the total volume. Conversely, if we break up this cube into smaller pieces, it's the same volume, but now much more surface area. And so our cells, or at least the cells in our kind of ancient lineages, developed this membrane system to create more surface area so that they can continue to get bigger without sort of collapsing in on themselves. And so the membrane system begins with the membrane around the cell. We happen to call that the plasma membrane. It is a bilayer of phospholipids, so 2 layers of phospholipids with proteins kind of studded within it. The phospholipids have a hydrophobic interior, 2 little tails that are afraid of water, hydrophobic, and then an exterior hydrophilic, water-loving head. The proteins are sometimes a channel, sort of like a tunnel for stuff to move in or out. They might be an enzyme, they might be like a protein pump that uses active transport to move stuff in and out of the cells. The plasma membrane proteins can have lots of different uses. So we have water, of course, both outside the cell and inside the cell in the cytoplasm. Because there is water both outside the cell and inside the cell, the surface of the membrane, both the outside surface of the membrane and the inside surface of the membrane, have to have those hydrophilic heads, right? Because they're water loving. The tails are hydrophobic, so they like to be away from the water. So they sort of hide out using the hydrophilic heads as a shield. And you can see here is a channel protein, and here you can see some other integral proteins that might be like a carrier protein or an enzyme or whatever the case may be. Again, very multifunctional plasma membrane proteins usually. So there are kind of 2 categories of cells. Prokaryotic cells, which belong to prokaryotes. Those are organisms that have prokaryotic cells. And then eukaryotic cells belong to eukaryotes, or eukaryotic organisms. All cells, whether they are prokaryotic or eukaryotic, they will all have a plasma membrane. They will all have some sort of genetic component, either DNA or RNA. They will all have ribosomes, which are protein-producing organelles, and they will all have cytosol, which is sort of this gelatinous kind of fluid that exists inside the cell. The 2 domains, bacteria and archaea, comprise the prokaryotic cells. These are, relatively small cells with relatively simple structures. Everything else, from the protists and the fungi and the trees and the flowers and the bugs and the humans and the whales and everything else that's not bacteria and archaea, are considered eukaryotes. They have eukaryotic cells. And what differentiates the eukaryotic cells from the prokaryotic cells is that they have a nucleus that is enclosed by another membrane, and they also have organelles that are enclosed by membranes. And you can sort of think of organelles as like the cellular machinery, what does stuff inside the cell. So here you can see a bacterium. This happens to be Helicobacter pylori, which is the bacteria that can very often lead to stomach ulcers. You can see they've got their cytosol, sort of the inside kind of gelatinous fluid inside the cell. No nucleus, instead they have a nucleoid, so much more kind of diffuse arrangement in terms of their genetic material. They will still have a plasma membrane, and you'll see they have a cell wall. Now, not all bacteria have cell walls, but many of them do. Some of them, in addition to a cell wall, will have these little capsules that help them like evade the immune system. And this particular bacterium has flagella, which are these little whip-like cellular extensions that do a little propulsive movement. But you can see here with both the diagram, and here is a bacterium, again, colorized using some computer algorithms, but this image was taken with a transmission electron microscope. It's nearly 21,000 times the size of the bacterium in actuality. So very, very tiny little organisms. The Organelles of a eukaryotic cell can generally be divided into kind of four kind of functional compartments. There's the genetic control of the cell, which is carried out by the nucleus and the ribosomes. There's how cells manufacture, distribute, and break down molecules. That's generally the work of structures like the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and peroxisomes. And then there are two kind of energy processing organelles, mitochondria, which belong in all cells, so both plant and animal cells, and then chloroplasts, which are found only in plant cells. And then last kind of functional group looks at kind of structural support, movement, communication, things like that between cells. And so we're talking primarily about what we call the cytoskeleton, the plasma membrane itself, and then the plant cell walls if we're happen to be talking about plant cells, not animal cells. So you can see here is a very basic animal cell. You can see here is its plasma membrane. There's its nucleus. The endoplasmic reticulum comes in two varieties. The rough ER, which has ribosomes attached, and then the smooth ER, which does not have ribosomes attached. Here is the Golgi apparatus, which functions sort of like a warehouse or a post office. Lysosomes and peroxisomes are sort of the kind of recyclers of the cell. The mitochondrion here is our kind of power plant, makes energy. And then you can see different cytoskeletal elements, things like microtubules, microfilaments, intermediate filaments, essentially proteins of different sizes that make up sort of a soft skeleton inside the cell. You'll also see something called a centrosome and some centrioles, which help during the mitotic process, so when a cell splits. And we'll talk about that in a later chapter, I think. Here is your kind of basic animal cell. So in addition to the plasma membrane, the nucleus, the rough and smooth ER, the Golgi, the peroxisomes and lysosomes, the mitochondrion, we have then this chloroplast here, also this kind of central vacuole, and then these little tiny pores called plasmodesmata in that cell wall, which again is a feature purely of plant cells and not to be found in animal cells. All right, we'll end it there, and we'll pick up the second lecture talking about the nucleus and ribosomes.